In today's webinar, we will talk a little bit about the need for modern informatics in gene therapy. Tom will follow that up with a live product demo, and then Jamie will present on how Verve is accelerating their R&D with Benchling. We will end with a final Q&A session. Please post your questions in the chat throughout the presentation, and we will answer them all during the Q&A. For those unfamiliar with Benchling, our mission is to accelerate life science innovation for the betterment of humanity. Our company was founded in 2012 by a team of scientists and engineers at MIT who were frustrated with the poor quality of software they had available to support their science. So with the support from top-tier investors like Andreessen Horowitz, Thrive, Benchmark, and Sequoia, Benchling built out a range of solutions for life sciences on a single platform. We are fortunate to work with over 500 innovative life science companies, including many of the world's largest enterprises. Gene therapy is at the forefront of developing breakthrough treatments. The number of therapeutic indications where gene therapy has utility is ever expanding, from genetic and CNS diseases to oncology and infectious disease. Second, the suite of technologies that scientists have access to innovate in this space is rapidly evolving. Examples include CRISPR and viral and non-viral delivery vectors. Finally, because of the tremendous potential of gene therapy, companies are bracing to adopt and scale their technologies to meet their R&D needs, especially in discovery and screening. I think this is a good opportunity to find out what type of research you are working on. A poll will pop up shortly on your screen. So the poll reads, what type of gene therapy are you working on? Is it and the options you have are DNA delivery with viral vectors, DNA delivery with non-viral vectors, oncolytic viruses, CRISPR gene editing, CAR-T, cell-based gene therapy, RNA delivery, or none of the above. We'll just give it a few seconds for people to vote before we close out the poll. Okay, great. So looks like majority of you are working with DNA delivery with viral vectors and CRISPR gene editing. Like those two seem to be the most popular among this audience today. So if we look specifically at typical teams working in this area, we see several common challenges. Molecular biologists have these newer tools for CRISPR design, but these are often not integrated with their existing software such as ELN and inventory solutions. This means there is a lot of manual back and forth between these systems. For cell line development or CRISPR delivery scientists, they are working on several different methods or workflows at once and need software tools that can handle both flexibly to accommodate different methods and structure to enable quick setup of multiple similar experiments. With an analytical scientist, the challenge is with stitching together data from multiple instruments, which is a highly manual and laborious process. Another poll should pop up on your screen now. So the prompt reads, what are the biggest R&D data challenges you or your team are currently facing? And the choices are tracing experimental context, manual and tedious tools, difficulty finding samples, painful to aggregate results, poor visibility to insights, collaborating with teammates, unnecessary communication, or updating data management tools. So we'll wait a few moments for people to respond. Okay, looks like tracing experimental context and like the manual and tedious tools are the two most common pain points across this audience. So we think every company and scientific team deserves a solution that will accelerate and support your innovations. When you think about the ideal traits that any digital solution should have in R&D, there are 4 core attributes to consider. First, it has to model the science you focus on, which means having the ability to build data models that fit your science. Second, it has to support the processes and workflows that your team performs and have the ability to scale, iterate, and evolve over time. Third, it has to empower users with data at every level, giving scientists insights about their experiments and team leaders data on their programs. So you can make both scientific and operational decisions quickly with complete and accurate data. Finally, it ensures that users will immediately recognize the value of the solution, see that it saves them time, and adopt it in a way that will help keep them compliant and consistent with the rest of the R&D organization. Let's look at each of these attributes a little more closely. Every CRISPR therapeutic has a variety of entities associated with it, such as DNA parts, genes of interest, guide RNA, nucleases, plasmids, viral vectors, cell lines, etc. Each of these entities have their own sequences, properties, and ontology. Every resulting gene-edited cell line has experimental results that need to be tied back to not just the cell line, but also the starting samples and sequences. So this is clearly a data modeling challenge, but it is also an operational challenge, since these also represent the day-to-day functions for lab scientists developing these CRISPR therapeutics. When we think of preparing digital infrastructure for these kinds of challenges, we have to think about both the data management and structure, as well as the operational considerations. Digital solutions have to be adaptable. The IT world has moved away from monolith software towards more agile approaches. But adaptable shouldn't always have to mean coding and application development. Many life science companies either don't have the resources to do this or want to use the resources they do have to focus on higher value operations. This slide here is an illustrative example of part of a CRISPR gene editing workflow, and it reflects the constant iteration and evolution that these specialized teams see on a daily basis. So in an ideal world, the data model, the process, and the workflow should be configurable in a low to no code environment so companies can iterate and evolve quickly without being resource intensive or overly dependent on their digital partners. Once you have the data model and process in place to capture the structured data, the next consideration is decision making. A modern digital solution should enable its users to make data-driven decisions. Different members of an organization have different questions that they need to answer and have varying decision support needs. For a scientist, this means uncovering insights across their experiments so that they can design, execute, interpret, and optimize the studies more efficiently without tedious data aggregation and data cleanup. For example, a scientist might be looking for an appropriate cell line to plan the study. Now, instead of emailing other team members or going to separate inventory and ELN tools to look for this information, the scientist should be able to look up and compare expression profiles and inventory data of multiple cell lines by querying a central data warehouse, where the data is already standardized during data capture. At the level of a team manager, the question that needs to be addressed are at the process and team productivity level. Now, typically, this information is collated and visualized with Excel or other specialized process management tools with separate data entry and reporting workflows. A modern digital solution should be able to connect the experimental and process level information in a single system, such that managers can track team productivity and throughput easily and answer questions such as number of outstanding requests, assay turnaround time, and view the latest progress on related experiments. Finally, at the executive level, you want a high-level view of your entire pipeline, the ability to compare candidates to make go-no-go decisions and allocate R&D resources efficiently. For example, the executives of a CRISPR therapeutics organization might want to look at key data across lead candidates to identify the best candidate to support major development milestones. Ease of use is the final important attribute of a modern digital solution. Most software tools have a learning curve, and then because of poor user experience, a forgetting curve. Only if the solution is easy to use can it ensure that users not only get trained, but ramp usage and realize the full value of the solution. An easy-to-use solution allows users to record experiments quickly, tap into the records to uncover the most meaningful results, and finally aggregate and analyze results across studies from the same user interface. This is essential to drive performance, boost productivity, and improve data integrity. Everything that I talked about just now is powered by the Benchling Life Sciences R&D Cloud, which is designed to give you a complete view of your R&D innovation. Benchling's applications span the R&D process from experimental design, in silico biology, and sample tracking, all the way through process management. There are 7 applications that Benchling offers, but they're all offered from a single interface. This makes it easy for users to leverage what they need without leaving the software. All of these applications are built on a common unified platform. They share a configurable data model that maps to each team's unique scientific and operation requirements. Benchling is built on a modern cloud infrastructure and offers state-of-the-art security and control. And we know that most companies have a rich ecosystem of instruments, software applications, and data stores that their R&D depends on. So we provide an open platform with a full set of REST APIs and event APIs and out-of-the-box connectors for lab automation, as well as ways to move data onto and out of the platform. Benchling provides a complete solution for gene therapy R&D, starting with tools that speed up guide RNA, DNA, and plasmid design to enterprise-grade tools that help optimize viral vector production faster and streamline the complex collaboration needs of in vitro and in vivo testing and reporting. With the introduction of Benchling Validated Cloud earlier this year, we provide a unified digital solution for both gene therapy discovery and development. Now you can utilize Benchling for workflows requiring validation, keeping the same approach to how you manage biological registration, data collection, and structured insights. It can also help R&D teams work better together by coordinating work across teams, bringing visibility to critical operational resources, program management, and scientific insights. With Validated Cloud, we provide additional capabilities such as isolated tenants with controlled product release process and application capabilities to help our customers maintain compliance, as well as the security and control needed to operate under strictly regulated conditions. We are proud to work with several leading gene therapy companies that have helped develop new advances in the field. This has helped build our domain expertise in gene therapy. With that, I would like to hand over the presentation to Tom to take us through the live product demo.
All right, thanks, Prem. So before we get started in the product demonstration, I'll just give you all a quick overview of what we're going to cover for today. All right, so for the demonstration, I'll be taking you through a few different scenes, starting with guide RNA design and bulk plasmid cloning, and utilizing these tools to create some new vectors where we'll cover their use and how I manage the guide RNA libraries within Benchling. And then next, I'll be performing a bioreactor run to produce some AAV delivery vectors, where we'll capture the run parameters as well as both online and offline data capture for those bioreactor runs. And then we'll round out by reviewing how requests are placed to the analytics team, along with how the analytics team manages those and executes on those requests. All right, so let's get started here. So here now we're in the Benchling platform, and designing guide sequences and connecting these sequences and designs with the corresponding experimental context can sometimes be tedious or slow. And being able to do that in just one place, as we'll see, is a way that can really tie these things together in a structured fashion. So within the notebook entry, which is our view here, I have a flexible workspace where I can both record those experimental results and also link them directly to that design. So here we're going to produce some guide RNAs within Benchling for a gene against a gene of interest. So here I have linked out this IL-6 gene. We're going to produce a guide sequence against exon 3. So because Benchling is sequence aware, simply clicking into that, it has a DNA sequence associated with it, and it brings me directly into the molecular biology tool of Benchling. Within this tool, we'll see the sequence. If I click on the map here, we'll see those annotations of those different exons along that gene. And if we look here, we have some mapped different guide sequences. So RNA oligos, essentially these spacer sequences that are aligned onto exon 1. For what we'd like to do, though, we'd like to produce them against exon 3, and I'll show you that design tool in just a moment. If we look a little at that data, the metadata associated with our gene of interest, these RNAs are actually linked directly to the guide, or the gene of interest rather, and are surfaced here. And clicking into one of these will bring me to that sequence. And because it's an RNA, it has the appropriate base sequences. But if I'm going to be producing this and wanting to chemically modify this, I actually have a place to record that in a really structured and visual representation of that. So within that, you see this tab over here where I'm looking at the nucleotide sequence, where I can then make modifications to the sugar or the 3' phosphate groups and track that within Benchling, those modifications. Moreover, though, if I ever want to share this or export it, you can simply click on this and actually copy the Helm notation, so I can maybe share it with someone else, or also configure this so that I can copy the IDT annotation for an RNA to get it directly synthesized. All right. So taking a walk back over to our gene of interest, though, here, we'll move, and I will. There we are. All right. So we'll first, to capture that new guide sequence and design that, I'll first click on our exon of interest here. So exon 3, and then move over into the integrated design tool. So within that, I'm able to come here and set the guide parameters based on my desired parameters. Once I finish that, then I'll come into the guide sequence, the design tool, which I'll do a quick shortcut and jump over here. So now we've seen here that Benchling has surfaced a number of guide sequences aligned to this exon 3. And it's also calculated both an on-target and an off-target score for me to use or sort against to be able to select a guide that I want to bring forward in my process. So we'll do that here. I'll just quickly select the top 5. And now I have an option to either directly assemble this into a vector of my choice within Benchling here, or export it into the registry so that I can maybe do a more sophisticated cloning. I'll do that here. So we'll export that out to our clipboard. And now we've captured all of these columns and rows of information into that clipboard. All right, so if I move into the registry, now we see already pre-selected here, I have the different other spacers that we have within the registry. And I can import these in bulk directly into my registry here with just a few clicks. And I'll paste in that sequence. And now Benchling will validate the columns match within the registry and those fields that we just captured. And then finally, I'll import them into the Benchling registry. So here I've imported them directly from the internal Benchling design tool. Conversely, you also, if you have your own internal tools for designing guides, you could do a similar function here to bring in a database or pull them in from outside sources. So because a lot of different tools tend to lack sequence awareness, it can be sometimes difficult to know what you've created and what hasn't been created. So when I just perform this action, Benchling actually is going to validate now these guide sequences that I just pulled in and confirm that they're a unique sequence here versus all of the others within the database. This really can speed things up or prevent you from making additional guides that have already been created in the past. So for our next step of the process here, I want to do a bulk assembly. So one way to move these parts out of my registry into the bulk assembly is to add them to a work list. So I'm going to do that quickly here while I'll add these into a work list for my spacers. All right. So now with that, we have our spacers selected. Now we need to kind of understand what scaffolding sequence to use. And one way to make a more informed decision about based on experimental information is to be able to surface this in a single place. So in order to support that, I've created I have a dashboard here that simply aggregates cutting efficiency, so something like an in vitro cutting assay of different spacer sequences with a variety of guide RNAs. So within this view, I see all these different scaffolding sequences and have guide RNAs that are roughly clustered around to give me a better understanding of which scaffolding sequences I might want to bring forward into my design. And if I wanted, I can also increase the stringency of this cleavage efficiency with a single filter. All right. And then getting back to our notebook entry, all of this information that we've just looked at, it's all captured here right within our notebook entry where we have our vector, our scaffolding sequences, an image of that result that we're basing our selection off of, and the vector of our choice, our backbone for cloning. All right, so now we're getting into the actual cloning aspect of the process here. So Being able to do that all in one place, we're able to come here to a bulk assembly tool where I have it mostly pre-filled out for our purposes here, but you'll note that I've selected a Gibson methodology. There's others available here, but Gibson will actually allow me to have both the in silico vectors, but also all of the oligos automatically created for me at the end of this process. So here I've selected the backbone. I'll then just quickly toggle in our spacer work list that we had just generated, as well as a list with those scaffolds that we'd like to use. So now that we've set up our Gibson assembly, we would then finalize, and in this one action now we're going to create 15 different plasmids and 45 primer pairs to perform this assembly. All right, so once created, the next step of our process really is to produce that vector itself. So we'll do that here and by jumping into a production run that's already in progress. Okay, so here's a production run that our team is currently executing based off of a request from the vector design team. So we'll get into how those requests are made and managed in a moment, but we see here at the top there that our input for this request, for this production run rather, is this transfer plasmid that they'd like us to use for producing this vector, this viral vector. Now that I have this context, I have both the plasmid, but also the full location and information on where to actually find this, which box in our inventory, and what container barcode I'm going to be looking for. So if we look a little bit more about what's being performed here, our team has registered a new virus based on that vector that's been requested, and are preparing for that bioreactor run. And they'll do so here through a number of steps, but one main step is formulation of medias. And having a lot of different recipes floating around the lab can sometimes be difficult to track. So having this centralized within Benchling can provide a real expedited way of doing this, as well as capture some of this information within the experiment. So here they've done that. They're utilizing a media recipe, which has defined QC criteria for it. And they've captured both the measured amounts for this formulation, as well as this pH, osmolality, and conductivity. Really, again, centralizing the capture of this information. All right, so next, really setting up these bioreactors quickly and completely using this information can be a large value. So they've defined that here, where the bioreactor run now is pulling from multiple areas. So the media that they've produced, the virus that was based on that cell line, or the plasmid that was requested, and they've defined some of these parameters for that run. Next, it can be a real challenge to view both online and offline data, and also capture it in a structured way in one place. So in order to streamline that process, we've actually utilized the Benchling API to do so, where our bioreactors are integrated directly to Benchling. And via that API, they're pushing at the end of the run that full data summary, which is going to have that minute-by-minute capture of the pH and the dissolved oxygen and the various parameters that those track, and all pushed directly into this one place so that we can better view it at a further step. At the same time as our reactor was running, though, the team is pulling off in-process samples, and they've captured and registered those here. And you'll note that automatically the virus that this was based off of is surfaced here to the forefront of this, so that anytime I'm interested in seeing a sample, I can know exactly the virus context of that, as well as the time points at which those were captured. All right. So, and lastly, after capture of these, they're going to capture their own offline data. So the OD, the amounts, the yields, and they'll do a quick titer on those before handing it off to the downstream team for HPLC purification. With this handoff, though, it's important to be able to present this information, this online and offline data, in an aggregated and stream in a consolidated place. And because we've surfaced this and we've centralized it all around the virus, if I click into the virus here itself, I see not only attributes of that virus that were captured, but also a contextualized dashboard that now has these 3 different bioreactor runs, summarizing both my in-process data, and then also all of those online bioreactor pH readings, where you can see that they've captured a large number of data points across just a short span of time here. And then all of this aggregated view is all captured automatically within Benchling. And this really can help to improve the accuracy of this result capture and confidence in the data capture itself. All right, so now at the end of our bioreactor run, they've handed it off at this point to the downstream team, which has purified it and submitted a request for analytical analysis. They did so by coming to the requests dashboard, where we can view current requests, but also submit a new one. So they've come here, submitted a new virus request, and filled in this form that's going to allow them to define which virus, prior lots, and click the wrong one there. It's this guy here, the analytical analysis. There we go. So things like availability data of the sample, urgency to communicate those things, as well as the contextual sample information. We'll jump into a request that's currently in progress, though. And we've been able to select it through filtering all of the requests that are in process. We'll look here specifically at this assay here, where we now have a summary of all that entire view, as well as those samples that need to be tested. Okay. So now taking the persona of the analytical scientist, they now are executing on their request in turn, and have all of that rich sample information, and now are able to perform their experiment, capture the cell lines that were produced on the back end of that, and then similarly capture results either in a manual fashion as are recorded here, or they can also do so using a direct upload of the output file from your qPCR machine. And they do so here by simply coming in here and dragging and dropping their files in, where they would import into Benchling and process that file, capturing all of that the defined information like CT values and sample within the sample context. All right. So hopefully here you've been able to see how some of these data are bidirectionally linked. And from our analytical viewpoint, we also have specific dashboards that they can view then to educate them on which kinds of delivery mechanisms and guides are the most effective. And all of these teams have done so in a centralized, streamlined way, where they're utilizing they're doing this data capture directly within their experiment. Okay, so now that'll conclude the product tour aspect of our presentation here, and I'll come here to just do a quick summary. So hopefully through this brief walkthrough, you've been able to get a better understanding of how Benchling can both save you time through integrating some of this experimental tracking and data capture within the experimental record. Also making the better informed decisions that are through integrated purpose-built dashboards, as well as increasing the ability to collaborate as a team through streamlining sample handoffs and request management. And then lastly, all of this sits within the platform that you didn't get to see today, but it all sits on top of a point-and-click configuration interface that can really keep up with the ever-changing needs of your science. All right, so with that, I'd like to quickly pause to poll the audience. All right, so what's the most important capability that you're looking for in a new informatics solution for gene therapy? And our answers are the same here as that was on our last slide: saving time, finding and analyzing your data, working as a team, and capturing new science. Just take another moment here. All right, so from the results here on the screen, it looks like most people are most interested in analyzing your data, and hopefully that some of that automated data capture that I showed you sparks a little bit of interest and curiosity about it, and how that could streamline some of your work. So with that, I'll hand it over to Jamie from Verve Therapeutics to give her perspective.
All right, thank you, Tom. And thanks to Benchling for having me here today. As an early employee of Verve, I was one of the scientists responsible for implementing the use of Benchling in our organization. And I'm looking forward to sharing with you how Benchling has supported our efforts as our company has grown. So our company, Verve Therapeutics, was created with the singular focus to protect the world from cardiovascular disease. We are a genetic medicine company with the goal to develop a single-course gene editing treatment. And we hope to do so by leveraging some of the great scientific breakthroughs of our time, including human genetic analysis, gene editing, messenger RNA-based therapies, and lipid nanoparticle delivery. So as illustrated in this slide, our R&D organization includes target analysis and discovery, mRNA design and development, formulation development, and preclinical assessment. Now, there's a lot of interaction between teams and commonality between how Benchling is used, but there are also certain features that have benefited each team individually. So, for example, my team, the discovery team, and the preclinical teams have been able to design several different configurations of results tables in order to efficiently report sequencing data. Now, the mRNA design team optimizes and modifies the mRNA, and as Tom showed, Benchling provides access to tools that can support those efforts. And our formulation team utilizes the registry and inventory system pretty effectively to track components of our different LNPs. Now, there are several challenges to being a small startup, particularly with regards to our data documentation and management. And Benchling was well positioned to support us through that as we developed into a larger organization. So Benchling provides a centralized location for our data so that those who should or need to see the data can do so with ease. At Verve, we highly value data integrity. And Benchling helps avoid the risk of introducing manual errors as data is shared and plotted by both scientists and team leaders. In a collaborative organization, it is important to mitigate against data silos. We've talked a little bit about that today, and support visibility of data across functions. For example, if I'm designing an in vitro experiment and I need to choose which guide RNA I want to use, Benchling enables me to access the analytical information generated by another team, allowing me to choose the lot of a particular guide that best suits the goals of my experiment without having to wait for a response to an email or to track someone in the lab down. And this slide illustrates some of the features that we have found Benchling does particularly well. So first, Benchling provides a relatively intuitive interface that new employees at our company are able to use quickly and correctly, allowing them to get started at the bench quicker than they might have otherwise. Also, the entity registration database that we've developed provides new employees a means by which they can envision how all the different facets of our R&D organization are connected and integrated. Next, Benchling provides a centralized database for our results, and it provides links between that data, which helps us through the use of dashboards to easily zoom in and out of our results when we need a high-level overview rather than just viewing the results for each experiment. So bottom line, we found that Benchling is well suited to support the efficient and effective collaboration in a fast-paced growth environment, such as at Verve Therapeutics. And with that, I'm going to hand it back to Prem for audience Q&A.
Thank you, Jamie, for that walkthrough of how Verve is using Benchling and just gene therapy R&D at Verve overall. I think that was very informative. I think at this point, like I see a few questions already being populated. I would request both Tom and Jamie join me on stage so that we can start answering some of these questions. So we have the full panel here. So yeah, if you still have any questions, feel free to add them into the chat box, and we'll tackle these questions just in the order of the votes the questions have received. So the first question I see on here from Michael is, can external tools be integrated into Benchling? So example is like implementations of new design algorithms. So Tom, do you want to take that?
Yeah, sure, certainly. So that's absolutely possible. So we have existing customers right now that have their own design algorithms and have integrated that successfully with Benchling so that there can be an easy crosstalk between the two.
Yep, great. And just to add to that, right? I mean, so when we talk about integration, beside algorithms, we offer Benchling for lab automation. And I think Tom mentioned that as well during this product tour. So you can actually integrate directly with analytical instruments and equipment to bring data directly into Benchling. So the next question I see is from Vega is, does Benchling have a way to automatically gather raw data from instruments and add it to the data warehouse? I think this is just related to what I answered, but do you want to add anything to that, Tom, to this specific thing?
Yeah, I mean, I showed you a real high-level brief overview, but utilizing the robust REST API that Benchling has, you can easily push things into the data warehouse either through a direct integration with. I had my example was like a bioreactor, but it could easily be any kind of analytical instrument to push that data to Benchling. Also on the other end, in terms of Benchling communicating outward, we do have an event platform to trigger things through AWS Bridge.
Yeah, and that's one of the newer ways we offer integration into Benchling, the events APIs. Yeah. Great, thank you, Tom. There's a question from Kiming Chu. If we can review the presentation again after the meeting. Yes, absolutely. I think as part of the post-webinar follow-up, I think you'll get email with the access to recording. And even if you haven't registered for the webinar, this webinar will be hosted on our website. So you can go to the event section to find this webinar as well as any of the past webinars that we have done. There's a question from Balaz Bogos. Is your database maintained locally, or do you rely on Benchling cloud service? Does that mean that you must be online 24/7? Tom, do you want to take that?
Yeah, sure. So Benchling is hosted in Amazon Web Services, so we're entirely cloud-based. So that does mean you need connectivity to access Benchling, so yeah, I mean that's part of the offering.
Yep, exactly. So we have 100% cloud, so everything, including data warehouses, in the cloud. There's a question from Richard. Is there a preferred browser to use? Had some frame shift with Firefox. Tom, do you want to take that as well?
Yeah, I can. I mean, I don't know necessarily if we don't necessarily recommend a specific browser. I would say that, personally, at least I use Chrome. That works quite well. I mean, I know others use Safari and things like that, but generally speaking, that's what I would use.
Yeah, I think that's what I've seen most of our users, both internally and externally, use. Chrome and Safari are the two more common browsers. There's a question from Joan. What does a data model look like? That is, what are the dependencies between the different entities in Benchling? Is this something that the customer has or can establish, or is this predefined? Tom?
Yeah, that's a really, really good question, right? So There are standard best practices that when we implement any new system that our customer experience team will help you with as a starting point. But that said, these interactions, it does serve as a relational database. So these kinds of there's parent-child type relationships, required fields. There's a wide range of configurability, and really allows you to model both things that are physical, like a tube of something, or more conceptual in nature, like what is my experimental conditions, and I'm going to capture that within a more custom object. And this is established at the front end of any implementation, and due to its nature, any admins or power users within your organization will have the ability to quickly modify these things. So just as a brief example, if I wanted to, let's say, add a field to my cell line to track passage number, I probably can do that in about 30 seconds. It is quite easy to add and remove things like that, if you have the accessibility to it.
Yeah. Maybe I think if Jamie can add on as well, what has your experience been in developing the data model for Verve?
Yeah, as we kind of set up our database, Benchling provided a lot of feedback with different links and kind of how we were envisioning setting it up, but that we designed it that suited our needs was one of the advantages. So the way that our entities work together, a simple example is that we have our guide RNA, our mRNA going into an LNP, and that every time we refer to an LNP, the metadata associated with all of the other components are linked. And what that metadata is can keep growing and growing, which is an advantage. So it's been very flexible, very easy to use for us.
Great. Thank you so much, Tom and Jimmy. So next question we have from Yoko. Do you see Benchling can replace Access database in data management, including curation of records, etc.? I think, so Tom, do you want to take this question?
Yeah, so we have a wide range of implementations within our customer base, and a lot of experience implementing things in a lot of different structures, right? So for larger organizations, a lot of times they'll have established databases, internally managed databases, and and they just need to recapitulate that within Benchling for the usability of Benchling. So that's always possible. So it's really on a case-by-case basis whether the Benchling registry, which serves as the Benchling database, is your only and single source of truth, or if you're pulling from other places and any kind of combination of the like.
Yeah, and maybe I'll just ask the same question to Jamie. So does Verve use the data warehouse, and if yes, can you share any thoughts there?
We do. We don't use it for everything. We have the data warehouse records, we use it for our workhorses. So we have entity links, results tables. We upload things with the run feature for large data sets. But we've found that it's pretty seamless with, other cloud-based systems in which we store and record data.
Great. Thank you, Jamie and Tom. So there's a question from Keeming, to whom we should contact if you want to do things like gene editing. So we have a slide at the end, it just talks of how you can request, if you're new to Benchling, how you can request a demo and get rolling, or if you're an existing customer, how you can approach as well. We'll get to that. I see one other question from Ming Chao. What is registry? Why do you need to register some items in your project? Maybe Tom, maybe you can start off, and I can add, and I'm sure Jamie can share her experience as well.
Sure.
I think this is a question we get often, I think, because people are used to just hearing of inventory, inventory solutions, among competitors and whatnot. So this is a question that's pretty common. Do you want to take it, Tom, first?
Yeah, I mean, registry broadly is used to it's just sample tracking, but also tracking of other types of information. Yeah, I kind of spoke to it a little bit. Conceptual things like DNA sequence, or more physical things like a lot of a virus or a lot of a plasmid. That sort of thing can also be tracked to and linked with the inventory tracking.
Right, exactly. And so just to add, right, the way I view registry is almost like the central nervous system, right, that connects all the samples and entities that you're working with. And it's like the theoretical entity, right? So whether it's like a DNA sequence or a cell line, there is the theoretical entity, and then there are the actual samples you're generating. So tracking the containers that have the cell line, the freezer storage location, like all that information is captured in the inventory, but you need a registry to kind of tie all these disparate samples together, right? So that's what helps you see, okay, like where did the cell line come from? Like, what was the plasmid that was used, and what was the DNA part that went into the plasmid? Like all of those, the lineage tracking and the context, the rich context that benchling provides is really possible because of registry and how it links these theoretical entities together. And maybe like Jamie, you can add your experience working with the registry and inventory.
Yeah, the registry allows our team to kind of connect all facets of what we're working with. So information about a particular lipid goes into a formulation that goes into an animal that turns into genomic DNA that gets sequenced. It allows for data integrity so that as you're moving through the different processes that function within our organization, there isn't translational errors, information isn't lost, and it provides, just that greater viewpoint of every facet of the experiment without having to manually and potentially incorrectly transfer and record that information.
Great. Thank you, Jamie and Tom. We have one last question. So if you can, the link to the presentation, yes, we will send that out as part of the post-webinar follow-up. So if you're registered for the webinar, whether you attended or not, you'll get the link to the webinar. I think those were all the questions we had from the audience. Let's see. I think we can. I think I would like to. Let's see. If there isn't anything else, should we just wait for a minute? Okay. I think I did have a couple of questions for this panel as well. Like just giving some time for people to ask any more questions. So I think I wanted to. I know Tom, you presented like the workflow with the viral vector workflow, and then I know like Verve and Jamie, like you are mostly working with non-viral vector workflows. So maybe I think we go to discuss like how Benchling can handle like both. So maybe Tom, can you just start us off, and we can get some more color from Jamie as well there.
Yeah, absolutely. I mean, the way that we typically would think about how to model these things is putting some extra flexibility in how you would capture experimental information, right? So whether you're performing your experiment using a viral delivery mechanism or a lipid nanoparticle, these things can be tracked in the same types of entities, so that from a workflow perspective, it's really just more about what the input is and how things are linked back in the lineage. Yeah, Jamie, I don't know, what are your thoughts there on how you guys do things?
So we primarily have just leveraged the registry. We've kind of created our own systems within that to relate different parameters to actual production and actual materials. I think that's as far as I can get into it.
Yeah, absolutely. No, thank you so much for that, Jamie and Tom. I see like one last question. So from Philip asks, are there any features to help with tech transfer? So Tom, do you want to?
Yeah. So when it comes to implementation, so if you have an existing, I mean, a good simple example is you have freezers and fridges at your any location, right? So there's a lot of bulk tools to upload those structures and hierarchies into Benchling in a bulk fashion. Same for things like if you have just even through the UI, like a large database of plasmids and vectors, those can all be pushed through CSVs. So virtually everything you do in platform, maybe through that you saw with the notebook, was kind of maybe a little bit more of a one-off fashion, can be done directly through the registry in a bulk way. And that really is what accelerates that time to kind of transferring things over, especially if you're using some other systems.
Yeah, and I think so. Just to add there, I think the question, like the tech transfer, is actually referring to moving from research to development, right? So there's tech transfer where sometimes research organization, like they're done with the research and they want to pass over the data to maybe STDMO or like maybe their own development teams internally to start scaling up. And sometimes, yeah, so tech transfer is go from research to development or from development to manufacturing. So I think the way I think I did briefly touch upon like Benchling validated cloud. So that's an offering where we are providing like a controlled, validated environment of Benchling, so that development teams can operate the same. So in the same not the same environment, but the same on Benchling. So because your research is on Benchling already, and now your development organization also is using the validated Benchling platform, it's easier for you to like share data and like transfer that context, from the research side to the development partners. And I know we have like more features coming down the line there as well to further streamline this process and really help you do your entire gene therapy workflow end to end on Benchling. So I just want to give a huge thank you to the speakers today, so both Jamie and Tom, for taking the time and like sharing your insights. I think it was very helpful. Even to me, actually, right? So I'm sure the audience enjoyed as well. And I want to thank the audience for joining us today, and to those who submitted questions. So please reach out to us with any questions. As you can see, if you're a new user, you can go to this link to request a demo, and somebody from Benchling will reach out to you. If you're an existing user, you could still request a demo, or you can contact the Benchling customer rep that you are used to working with. And then we have a lot of content and webinars and events happening all the time. Please visit us at this link here to look for upcoming events. So a closeout poll will just pop up. So to tell us if you found today's content interesting and useful. So please take a few moments to just rate this webinar. It will be really helpful to us to improve in the future. Yeah, thank you so much. Thank you again, everyone, for joining us today. Take care. Bye-bye.